Good afternoon. The doors are closed. You are locked in. That is my cue to start. My name is David Yokum. I have the real pleasure of introducing Lynn Nadel and being part of kicking off a celebration of his receipt of the APS William James Fellow Award, which is for a lifetime of significant intellectual contributions to the basic science of psychology. Let's begin, as all proper odes to a scholar should, with a haiku. If you would please close your eyes, relax your shoulders, let Lynn sit above you. Remember this fact: Lynn's stature in the field, a campus-sized hippo. I didn't promise it would be good. So, for those of you who know Lynn, of course, he's for almost half of a century. Lynn Nadel has helped to set the international research agenda in studies of the hippocampus, spatial cognition, and episodic memory. His 1978 book, The Hippocampus as a Cognitive Map, co-authored with John O'Keefe, drew on a careful review of decades of studies and laid out a comprehensive vision for how the hippocampus learns and stores cognitive maps of space and context-dependent memories. It was initially met with a little bit of skepticism, but this idea over time has drawn support from a substantial amount of other research, including the discovery, for example, of head direction and grid cells in the brain that help animals navigate their environment, as predicted by cognitive map theory. Lynn later advanced multiple trace theory in collaboration with Morris Moscovich, which holds that the hippocampus is always involved in storage and retrieval of episodic memory, but that semantic memory can be established in the neocortex. Lynn has also investigated fundamental aspects of spatial learning and cognition, the impact of stress on hippocampal function, and the contribution of hippocampal dysfunction to intellectual disability and Down syndrome. His recent work on memory reconsolidation, spurred by multiple trace theory, has led to new ideas about managing enduring change in psychotherapy. And speaking personally, and if you've ever interacted with Lynn, you know this, and I've known him for a good decade now, beginning as his student at the University of Arizona, then you'll know that he's really also just a phenomenal teacher, mentor, colleague, and friend. You taught my favorite class in graduate school on the history of psychology. I actually still have some of the essays that I wrote to you, including your notes back to me, if you can believe it. And Lynn was personally pivotal and encouraged me to take a risky career leap that he described, and I quote, "almost sure to fail, but still worth it, because of how unique the opportunity to learn was, and how unique the swing for big social impact." And I think it is that spirit of scientific adventure, that bold creativity, and also that humble wisdom of thinking about science in its most technical terms, but also the meanings that it has for our everyday lives, that makes him the sort of scientist that I want to emulate and that I think we all deserve to celebrate. And so, Lynn, thank you, and congratulations on this well-deserved recognition.
Well, that was a very humbling introduction, actually. I'm almost certainly going to fail to meet the expectations that has created. And I'm also going to fail to meet the expectations that my title may have created, because you can take James seriously in many ways. But before I get into the talk, let me thank the organizers of this. Let me thank APS for thinking I'm worthy of this sort of an award. All of those who've contributed to my success over the many years, including my friends, my colleagues, my spouse, many people. Thank you all. Again, very humbling. Now, taking James seriously. I don't know why I chose that title. Partly because I gave a talk at APS last year where I talked about all of my current work, so I was like, what am I going to talk about? I chose this because I thought it would be interesting to actually go back and think about James in the context of some of the earlier work I had done. And that's what I'm going to talk about today. So the thing about James that I'm going to take seriously is, of the many things one could take, is that he understood that there were different kinds of memory. Right? He introduced that idea that there were different kinds of memory to the field pretty much although there were inklings of it prior to him. And he coined the difference between habits and memory. He didn't talk about them as multiple memory systems, but he was clearly talking about different kinds of memory. So that's the part of him that I want to take seriously. The other person I want to take seriously, I can move away. Right. And I can use this. The other part I want to take seriously is, the other person I want to mention is Don Hebb, who was a mentor of mine. I was at McGill as a graduate student, and he sort of picked up in some sense where James left off, buying into the notion of short-term and long-term memory, but added issues around brain plasticity that James had also thought about and connected them with his theory of cell assemblies that made the whole thing make sense. And so the field expanded with Hebb's building on James, and I want to it's not humble at all to say I'd like to continue with that. And this is not working because it's not on. Okay, the lead actor in this story is the hippocampus. There it is, and you can see it in a couple of different pictures. That one over there shows you why it's called the hippocampus, because it looks like one. Right? And this, I think this should, well, doesn't work. That picture there of the seahorse, that should be animated, but it's not going to happen now. Anyway, it looks like a seahorse. The anatomist who sort of extracted the hippocampus from the rest of the brain saw the similarity. They called it hippocampus. That's why I don't want to waste too much time on that. The modern era of hippocampus started with HM. There he is. We now know his full name, Henry Meliasen. He was operated upon by this neurosurgeon, Scoville, in New Haven, for relief from impossible epileptic seizures. He simply was seizing all the time. The surgery, which was experimental at the time and is described in a variety of books, was to remove large chunks of the medial temporal lobe, which is where the seizure focus was. They did that. It was successful in kind of mitigating the seizures, but it had a devastating effect on his ability to form new memories. Right? So the history of how that happened is that Scoville, having made this surgery and having noticed that there were some problems with his patient, he got in touch with a friend of his, Wilder Penfield in Montreal, and said, are you interested in working with this patient? And Penfield got in touch with his friend, Hebb, in the psychology department, and Hebb gave it to his graduate student, Brenda Milner, and that's what she looked like when she was a graduate student in 1950. This is closer to what she looks like now, but actually she's now 101 or 102 and still going strong. Amazing person. Brenda gave it to her student, Sue Corkin, and Sue spent her entire career studying HM, including writing this very excellent book about HM that I highly recommend. A little known fact that some of you may be aware of, she married my college roommate in 1961, and that's when I first heard about HM. Was from Sue Corkin in those days. By the way, that marriage didn't last. A random fact. That's for Sue's benefit. All right, so in understanding what happened with HM, you had to understand what the field thought about memory at that time. Right? So this is the way people thought about memory when when the data on HM came out. The idea was that something happened in the world, and there was some sort of an immediate memory that was formed as a consequence of that, like iconic memory or echoic memory, and then you would attend to some portion of it, and that would go into short-term memory. There would be this short-term memory buffer, and that would last for maybe 20 seconds or so. Some of that would be forgotten, some of it would be rehearsed, and some of it would be consolidated into long-term memory. Right? That's the basic story. This is the Atkinson and Shiffrin model, more or less. And by the way, there was just recently some journal, I think it may have been Cognitive, it was a 50-year anniversary of the publication of that Atkinson and Shiffrin paper with all kinds of discussions about how influential it's been. But this is only part of the story, is what I'm going to tell you. So within this story, you have multiple memory systems, so to speak, but they differ in terms of how long they keep the memory for. They differ in terms of the duration of the memory. That's the main difference between them, right? This one lasts a very, very brief time. This one lasts a little bit longer and can be rehearsed. This one lasts as long as anything lasts, all right? Now, HM added to this picture because HM apparently had intact short-term memory. You could say something to HM like 574, and then wait a little while and ask him to say it back, and he would be able to give it back if you didn't distract him. Right? As long as he was undistracted, he could keep things for up to 20 seconds. So that was intact. It also seemed to be that his long-term memory was intact. They would ask him, well, can you tell us something from before the surgery? And he would give them a big, long story, and okay, his long-term memory is intact. Maybe there's a problem for a year or two. That was the initial finding. Well, if that's the case, but there's this complete inability to get anything from here to here, which is what was being reported at the time, there had to be a problem here. So the theory was that as the surgery largely took out the hippocampus, this part of the brain was assigned some role in memory consolidation, and that was where HM led to. All right. The problem immediately was that the same people in Montreal then tried to create an animal model of exactly this dense amnesic syndrome that HM was demonstrating. They took monkeys. The same surgeon, Rasmussen, was at the Montreal Neurological Institute. The same people made what they thought was a comparable lesion in these monkeys to what HM had suffered, and then they exposed the monkeys to a set of learning and memory tasks, and guess what? No problem whatsoever. Zero amnesia. The field was thrown into complete chaos. I think last year I showed a picture of some people fighting. It wasn't that bad, but it was complete chaos. And the problem was at the time they simply were thinking wrongly, not good English. They were not thinking correctly about how memory is organized in the brain. That's my view. All right, everything stayed that way. Let me go back. I was a graduate student at this time. Right? I said, those years, I became a graduate student at 65 or 64. And I did my thesis trying to resolve this issue, by the way, and completely failed. I did get the thesis, the PhD, but I failed to make a dent on the problem. The problem remained a problem for the field for about 10 years, and then this came along. Then John O'Keefe discovered place cells in the hippocampus, and that changed the conversation about what the hippocampus could possibly be engaged in. Now, I got involved in this for kind of crazy non-scientific reasons. I knew John. He had been a graduate student at McGill as well. We were good friends. He took a postdoc in London. I took a postdoc in Prague in 1967. Then that happened. You may not be able to see very well, but down in the corner are pictures of the Russian invasion of Czechoslovakia, where I was living, where tanks came up the street. I left Prague, went to London, and at that point cooked up a deal to go back to London to work in the same lab as John was in, not with him. And then I was lucky enough to be there when he discovered place cells. And he came down the hall and said, you worked on the hippocampus in your dissertation. What do you think, doesn't this look like spatial stuff? That was it. I dropped what I was doing. No longer interested in the vision. Sorry. My wife does vision. And then I shifted into the hippocampus. And then over the course of the next 7 or 8 years, we wrote that book. And the book was a vast compilation of everything that people thought they understood about the hippocampus and what we made of it. And I can't make anything of the fact that I was wearing sunglasses at the time. That book largely was interpreted as telling a spatial story about the hippocampus. And many of the things that happened subsequent to the publication of the book focused on this is a spatial system. What people didn't exactly cotton onto was that we were really talking about memory all the time. Right? So here's a quote, not from page 423 in a footnote, but on page 1 of the book that says, it's the core of a memory system. Right? So that was kind of ignored, that we were really talking all along about memory, but as David pointed out, context-dependent memory, the crucial point. Right? So given that approach, the cognitive map theory has some implications for memory. One of those, so these are quotes that were from the 1970s. One of those implications is that memories are used to predict. So we were into the notion of memory as generating predictions right from the start. Cognitive maps predict what you might expect to find around the corner, so to speak. The second implication, there are multiple memory systems in the brain. It's not, here's again the quote, it's not implied that all memory is stored in the hippocampus. Memory systems concerned with lots of other things, values of things, motor habits, etc., they're all outside the hippocampus. So this was the foundation for the idea that there are multiple memory systems in various parts of the brain doing different things. And this is the different things part. The memory systems vary not just in duration, as the Atkinson-Shiffrin model had talked about, but also in content. Different memory systems deal with different content. That was the claim. Right? There's no such thing as the memory area, a la the medial temporal lobe memory story, there are memory areas, each one of which is responsible for a different form of information. So the point here was that the hippocampus was claimed to be responsible for this kind of information, particularly. All right, the hippocampus, that's our focus. The assumption was that it represents information about space and episodes. Oops. And that it supports behavior dependent on contexts and places. However, and this is what the rest of this talk is going to be about, those first three things are what I talked about last year, if any of you were here last year. The thing I didn't talk about is the other claim, which is that memory systems have distinct developmental trajectories. These different memory systems, which have different properties and are concerned with different information or kinds of information, have different developmental trajectories. And that turns out to have very interesting implications, and that's what I'm going to be talking about now. So here's a rough figure, gives you the sense that certain kinds of learning and memory are sort of available to the organism early on in life, and others come on later. Here you can see the claim that the hippocampal form of learning comes on later. What that reflects is the notion that actually the hippocampus, amongst various brain structures, has a postnatal maturation story. The hippocampus is not intact and functioning in most species at the time of birth. There are a few exceptions. They're called altricial species. Guinea pigs are one of them, and ungulates are another, because you know, like deer and antelope, when they come out, they're ready to go right away, so they need a hippocampus. But most animals are not ready to go right away. Hippocampus develops late. In rodents, this is roughly the story. There's lots of different indicators of things that show up after birth. P is postnatal. So neurons are being formed before birth, but they're also being formed after birth and actually for the rest of their life. There's this neurogenesis in the hippocampus. I may get back to that. Synapses are being formed pretty much a long time. A lot of other physiological and anatomical measures are only showing up postnatal 2-3 weeks. And various functional indicators indicate late onset of function as well. So that's the basis of talking about hippocampus as late developing. It's also true in monkeys. If you look at the growth, these are different parts of the hippocampus, but the growth curve is shown this way. This is a slow increase in all of the parameters of hippocampal function in monkeys from life. If you look at humans, it's the same thing, and there's a dramatic increase in the size of the hippocampal formation in humans and the anterior temporal lobe in the first 2 years of life. So the point about hippocampus then is that in humans, its function seems to emerge in the sort of 16 to 24 month range. Right? There's very little evidence of hippocampal sort of functionality before that. Indeed, the development of hippocampus probably continues for quite a long time, but I'm not interested in that adolescent period. The point I want to talk about now is, given this story, what does it actually mean to grow up or to start life without a hippocampus? How does that affect how you learn? How does that affect how life proceeds, so to speak? Three kinds of effects that I'm going to talk about in the talk. First of all, there are the direct effects of not having a hippocampus, which means that whatever it is the hippocampus is crucial to do, those animals can't do it, or those people can't do it at that age. That's the first type of problem, so to speak. Then there's an indirect effect of the absence of the hippocampus. That's not me, right? The indirect effect is that all the learning that happens at this stage of life is happening in other brain structures, in other learning systems that have different properties, some of them. Therefore, the learning that is taking place pre-hippocampal may have different properties than the learning that takes place post-hippocampal, at least in its totality. A little bit of Freud in that, by the way, which we may or may not come back to. The third way in which this late developing structure matters is that because the system is still developing, it may be uniquely susceptible to sort of perturbations that come in either from the environment in the form of stress or various toxic things, or to chromosomal abnormalities may play out in a different way in structures that are sort of more pliable, so to speak, in terms of development. All three of those are possible implications of having a late developing structure like the hippocampus. And I'm going to tell you something about research in each one of those things. That's basically the talk. All right, so the direct effects. I should look at the time. How am I doing on time? There's no clock there. 1:20, and I have until 1:50, right? I'm good. All right, so the direct effects of the absence of the hippocampus. I'm going to just talk about two. This first one here, I don't have any data. I'm just going to show you some data. And then I'm going to show you some data from these. So I did a lot of these experiments back in the 80s and 90s when I was focusing on development. So this is a sort of a rehash of some things that I haven't thought about for a long time, but nevertheless care about. All right, this inability to form episode memories early in life. That's a classic. These are some graphs from David Rubin. And the point is that no matter how you measure it, you see essentially the same thing. Males and females, whatever age you measure it at, however you cue for it, you pretty much see the same thing, which is virtually no episode memories reported before the age of 2, no matter how you look at it, all right? For those of you who are old enough, that quote should bring a chuckle. So why do we have this? And there are kind of 3 general theories out there. One is Freudian, you know, that you actually formed memories very early in life, but you repressed them. The other is that everything you learned early in life, you didn't learn it in a way that you could verbalize it, so that there's a non-translatability problem. And then there's this biology problem. They may all be true, by the way. These are not mutually exclusive. I focus really on this one, that a large part of the problem has to do with the late maturation of the relevant structures, including the hippocampus. Some people have begun to talk about the high levels of neurogenesis in the hippocampus as partially responsible for infantile amnesia. I don't want to get into that right now, but that's an interesting side theory. Okay, so here's some data. So just the exploration, just the interest of an animal to show interest in exploring its world. When does that emerge in a rat, right? And mind you, the hippocampus in the rat, development is between 21 and 28 days. It begins to emerge anywhere from 18 to 26, in that general zone, maybe a little earlier in terms of some measures. So what we did was we put rat pups, little babies, in an environment where they could show us how interested they were in exploring objects in this open field. Right? This was a box. The box was then pulled up so that they were put in there in an enclosed box. The box was pulled away, and then they were confronted with an open field and a bunch of objects. And we're just interested in when would they start to explore the objects? That was the question. And the answer is these are individual rats. How many rats started to showed exploration of any level on these various days? And the mean is around somewhere between 20 and 21 days that they first start to show interest in objects. Right? Now you might say, well, does that mean that that any given rat shows a little bit of interest here and a little bit more interest there and so on? Is it something that emerges gradually? The answer is no, interestingly enough. If you sort of shift the data so that you simply take the moment at which any given animal shows exploration and then you look back to what they did the day before, it was virtually nothing. It's an absolute phase transition for any given animal. On one day, they are showing no exploration, and the next day, they're at max. So whatever it is, there's a switch. There's some threshold, and this system then is going with respect to exploration. So that's an interesting finding with respect to exploration. We showed the same thing. We tried to show the same thing in a study with humans. So this is a kind of a version of the Morris water maze. Do people know what how many people do not know what the Morris water maze is? Okay, good. A task where you can sort of figure out whether people know where they are in space or animals know where they are in space. This is a 2-meter wide platform. This thing, the gray circular thing, there were a bunch of pie plates of the same color surrounding around the perimeter, basically. And under one of them, the kid was brought in to the space, allowed to crawl in, and then they saw the experimenter hide a toy under one of the plates. The plate was put back, the kid was brought out, the kid was brought in again and had to find the plate. And in this situation, where these cues are far away, and there are no differentiating cues in here, it requires distal use, requires the hippocampus. 1-year-olds fail at it, and 2-year-olds succeed at it. Right? So that's in humans. That's the measure of an exactly similar task that does not require the use of the hippocampus. That is, there are proximal cues, one of which is right near the appropriate one. The animal can just go to this one. 1- and 2-year-olds succeed at that. That's a non-hippocampal version of the same task. So this is a demonstration, as it were, that you need the hippocampus to solve this kind of a task, but it's because of the need to be able to map space in a certain way, not because you need to be able to remember that something's under a pie plate. This shows you that the kids, even at 1 year old, can do the task. They just can't use this kind of information to do the task. All right, so that's the end of part one. That sort of more or less conveys that, yes, indeed, there is a direct absence of memories and spatial learning and exploration while the hippocampus is developing. So that's implication one. Implication two, I'm going to skip over the indirect thing. I'm jumping to the susceptibility one. I'm going to come back to the indirect one if I have time. Environmental stress. Why would we think that environmental stress should have a particular impact on the hippocampus, on the developing hippocampus? There's a reason. That's the reason. In 1983, roughly, which was when I was beginning a lot of this work, Bruce McEwen and others began to report that the hippocampus was loaded with stress receptors. So now you had this structure in the brain that everybody was talking about as a memory structure, and suddenly somebody comes along and says it's loaded with stress receptors. Okay? That's a whole other story, which I will not tell you much about today, but one of the implications is, given all those stress receptors and a developing system, what happens if you stress the animal? Is that going to change the development of the hippocampus in some interesting way? Right? So here's what we did. We reared rat pups ourselves. We took those rat pups, in 2 different procedures, we basically stressed them during the first few weeks of life. We had 2 different ways of doing it. One of them involved isolation from the dam in a heated cage. Another one involved shaking them in a box. I'll show you. We kind of created this shoebox-like thing, and we put the pups in there, and then we shook it around for a few minutes. So we did various things to stress them. Right? And then we asked, okay, what is the impact of that stress on the emergence of these various things that the hippocampus is responsible for? Right? So this is to remind you, this is what happens in the normal case. These are rats that are normal, so to speak, unstressed. And this is what happens in the stressed animals. The emergence of exploration, on average, moves several days earlier. Stress appears to actually speed up the maturation at some level of this system, or the emergence of this response, of the emergence of exploration. Now, you might say, well, that's a good thing, right? Not necessarily. Development, of course, is many things are developing simultaneously, and the synchrony and timing amongst those things is important. If you pull something out of that structure and mistime it, so to speak, that's not necessarily a good thing. We were uncertain at this point. All right, what happens with LTP? We looked at LTP in slices taken from the rats who had been subjected to this. And what we saw was that actually the rat pups who had been stressed showed more LTP, more synaptic plasticity than the rat pups that hadn't been stressed. That's what this shows you. Right? This is the stress group. This is a measure of LTP, of long-term plasticity. These are the controls, and this completely befuddled us until we realized that developmentally, what actually happens is that developmentally, LTP starts out very high and actually comes down over the course of development because inhibitory circuits are filling in. So what this represents is a failure of the development of proper inhibition that actually tamps down the amount of plasticity. It's a funny result, but that's what happens with stress. What happens to behavior? So we took some of the same rat pups that had been stressed early in life, and we did this kind of T-maze task, the classic Tolman place versus response T-maze task. You know, in this task, you put a rat down here, and they run up, and how did I do this? They run up over here, and they get the reward. Now you flip it, and now they start from here. And if they go over here, then they are going to the same place. If they go over here, they're making a response. There are 2 different strategies, right? When they learned this task the first time, they go like this. They could have learned just go to this place, or they could have learned make a right turn. We don't know. You know by flipping the start arm. Now if they learn to go to their place, they go here. If they learn to make a right turn, they go there. Right? Did I get my right, left correct there? I have problems with that. Okay, well here's the thing. The place strategy, as you know, requires the hippocampus, but the response strategy does not. It is mediated by striatal mechanisms. Animals without hippocampus can do fine on this task. They just do it in a different way, so to speak. Okay, well, here's what happens. If we do this experiment with rats that had been stressed compared to controls and simply ask how many of them use a place strategy and how many of them use a response strategy, this is the normal effect. This has been reported in probably 1,000 rats over 50 years. Generally speaking, about 4 out of 5 rats prefer going to the place. That's the standard thing when they have an open environment. However, the same exact environment, the rats that have been stressed, the percentage of them now has dropped to 2 out of 5 there's a big impact on the likelihood of using this stressed hippocampus now to solve this task. So stress is actually a bad thing. Early stress, according to all of these measures, is doing something that has bad long-term consequences for the sort of normal functionality of the hippocampus. We see the same thing. This is very exploratory, very pilot. This is an experiment we did quite a few years ago, never got published, but I thought I would show it because the data were quite striking and it's interesting. Maybe somebody will take this up and think it's worth pursuing. This was a study where we looked at kids with asthma. There's a big asthma research thing in Arizona, so there were a lot of kids available. And we looked at the effect on these kids of taking corticosteroids for asthma treatment as an interesting group, right? And there is a massive impact in these kids on hippocampal. This is recalled statements as an episodic memory task. There appears to be a very big impact in kids who have been subjected to steroids for asthma, without any effect on IQ, by the way. Notice that there's no impact on this test. It shows up on the episodic memory test, so it's somewhat selective. Again, very preliminary, but indicative that you might see the same thing in humans who have been subjected to steroids early in life. There may be problems emerging in hippocampus, worth looking at. On to the chromosomal story. How many of you know about trisomy 21? How many don't know anything about trisomy 21? Trying to get a sense of how many. Okay, trisomy 21 is the causal condition for Down syndrome. You have an extra chromosome 21, right? And the implications of that means that you have one extra gene that is producing more gene product of all of the things that are coded for on that chromosome. One of those, by the way, is amyloid, APP, amyloid precursor protein. So that there is a link between Down syndrome and Alzheimer's disease that is very interesting and important. That's not what I'm talking about. Okay, there is this fact. So how do you connect it to the hippocampus? So here's a story. 1983 or 4, phone call. I'm so-and-so, you don't know who I am. I work on Alzheimer's disease. I'm putting together a meeting on Down syndrome, and I want somebody to come who can talk to me about the neurobiology of Down syndrome, about which we know nothing. And I asked Mort Mishkin for somebody who likes to speculate a lot, and he gave me your name. So that's how I got into Down syndrome research. And so I started to read the literature, and lo and behold, there was a reason for me to pay attention to Down syndrome, because it turned out that if you look at even what was known then, there were little fragmentary sort of bits of data. This came later, but there were fragmentary bits of data, and it turns out that the hippocampus is amongst the structures which is significantly smaller when you correct for brain size. This is not a brain size issue. Down syndrome kids have a smaller brain, but this is beyond that, right? There are other structures, the cerebellum, parts of prefrontal cortex, the whole brain, of course. But notice the ones that are sticking out here as disproportionately smaller, right? The perspective that I then developed was a hypothesis that late maturing brain structures, including the hippocampus, but also parts of the cerebellum, the neocerebellum, and the archicerebellum, prefrontal cortex, they are uniquely susceptible to sort of improper development in Down syndrome because of their late maturation profile. That was a hypothesis. And there would be a problem with inhibitory balance. I don't need to go there. And these structures, I claimed, this was back in the 80s, were most at risk in Down syndrome. And that general story has now been played out in the literature, and mouse models have been developed, and they all have problems with hippocampal LTP. And so there's sort of a grain of truth to this story, which remains to be fully explicated. But I want to show you what we did. So we went back and looked at this task again. And now the prediction would be, if they have problems in their hippocampus, then the 2-year-olds that succeeded on this place version of the task, right? This is the one that you needed to be 2-year-olds to do, because it needs the hippocampus, that they would have a problem with this task. And indeed they did. So these are toddlers who have mental age of about 3 or 4, right? And they failed this task nonetheless, right? Whereas they succeed on the non-hippocampal version. So again, it is not the point that they can't do this kind of task. It is that they cannot use that kind of information adequately to solve this kind of a task. So we then wanted to go on and test this sort of late developing sort of structures at risk hypothesis, but there was no way to do it. We did not have a kind of a battery for doing it, so we had to develop a battery that was sort of precisely targeted for the particular structures that we were interested in surveying, kind of. All right? So with my colleague, Jamie Edgen, we developed what we felicitously called the Arizona Cognitive Test Battery. And this is now being used kind of somewhat worldwide. And the point is that it has tasks that are sort of specially targeted to each of these three structures, as well as a bunch of benchmark measures. And the idea was to be able to point out that if we compare Down syndrome kids to mental age match kids, so that they're essentially of the same IQ, then any impairments they have are disproportionate impairments compared to their sort of mental age, you might say. And that was what we were looking for, and that's what we got. So we have a variety of tasks here. I don't want to bore you with all of this, but in each case, there was a task where there was a disproportionate deficit in the kids with Down syndrome, beyond what you would expect given their mental age. So there is sort of general support for the idea then that because the hippocampus is developing sort of throughout the first few years of life, that it is more susceptible to being interfered with. That developmental process is open to interference in a way that a fully developed structure might not be. And this is some of the evidence for it. It can be either stress. By the way, it's not just stress. We know that fetal alcohol syndrome, lead exposure, mercury exposure, chlorpyrifos exposure, which, by the way, is a DDT-like pesticide that was about to be struck from the list of usable things until recently. I can tell you more about that if you care, because I did some of the original work on that, on why chlorpyrifos shouldn't be. Anyway, the hippocampus is interfered with, its development is interfered with by a variety of such toxins in the environment. So it's a very susceptible. You can ask questions about why is the hippocampus so susceptible. That's an interesting cellular molecular story that I won't get into. That's the second part. Third part. Still good with time? Okay, this is the completely speculative part. So again, learning occurs in and reflects these already functional memory systems. In the first few years of life, when you don't have a hippocampus, the rest of the brain is doing lots of learning in various other systems. What are those systems? What are the properties of those other systems? What are they like? What is the learning that you do during those years going to be like, so to speak? Well, we all have intuitions. But the critical point that I want to emphasize is that because the hippocampus is linked to learning in contexts, everything you learn early in life is going to be context-free. It's not going to be sort of labeled as only do this in this context, or this works in that context. It's going to be only do this or do that everywhere. All right? So the context-free generalization, that's the major point that I want to focus on. So Jake Jacobs, and by the way, the WJ stands for William James, believe it or not. So Jake Jacobs and I, this goes back again to the 80s, but Jake and I are continuing to work on this right up to now. We thought that this fact, that the kind of learning that went on early in life was going to be sort of context-free, would generalize broadly, that this might be an interesting lever to understand some anxiety syndromes. Right? And that's what got us into this. So it turns out, as many of you know, that lots of anxiety disorders and dissociative states involve memories that are quite decontextualized. They're fragmented. This is a not uncommon way of describing what memory is like. And it sounds a lot like the kind of memories that are formed early in life. So we suggested, Jacobs and I, well, let's go to this quote first. Now, these are two names that very few people in the room might remember. This is Neil Miller. This is, I don't know what dollars. These were students. Well, Miller was not just a student. These were colleagues of Clark Hull. And so they were attempting back in the 1950s to apply conditioning principles to understanding things like anxiety disorders. Look at the quote. This is spot on to what Jake and I were on about. The young child doesn't notice or label experiences, right? Can't tell you about them, but the behavioral record survives. That's all about the survival of habits that have been acquired early in life, but which you did not name, which were not acquired within the context of an episodic system, right? So they were onto the idea. Anyway, we thought that this was an important insight that got lost, sort of, and that this might help account for certain anxiety disorders. So here's how it could work, and now this is pure speculation, pretty much. So early in life, you could say some aversive learning proceeds in the absence of hippocampus. Let's say you get bitten by an ant, or you see a spider when you're 1 year old. Let's take that classic kind of example. You get, this is a little Albert kind of experiment. You acquire that information, right? The claim is that you're acquiring it because the amygdala, which is necessary for acquiring any fear kind of learning, is fully functional at birth. That's okay, no problem there. But it is, again, not going to be context limited. It's going to be a general fear of that thing, or whatever you remember that thing to be, right? Again, the assumption was that these fears that you acquire early in life, because they're decontextualized, they're going to generalize very broadly in a way that is not normal, right? They create strong traces with no record of where or when they occurred, and quite frequently this is what is reported. So somebody with a spider phobia might well say, I have no idea why I'm so afraid of spiders, but I am. Right? That's not uncommon. All right? Similar things can happen in adults, it turns out. If you stress an adult, stress hormones get released. They sort of facilitate the function of the amygdala. But if the level of stress is high enough, because of all those stress receptors in the hippocampus, they shut it off. So under high stress, learning may be just like it is in an infant. You may be learning without your hippocampus under high stress, which means you're going to learn something a little bit different than you would learn under normal conditions. So we did some experiments maybe 10 years ago where we looked at, these were experiments with human subjects, looking at slides like the Dan Riesberg slide deck with events going on, and then asking the question of the ability of people to remember the neutral elements or the emotional elements in the story. Basically, that was the experiment. And what we saw, sorry for the small type in this one, I should have fixed that. There's an enhancement of the emotional elements and a decrease in recall of the neutral elements. Our assumption is that the amygdala is crucial for the emotional side of the story, and the hippocampus, which is encoding the contextual background, is crucial for the more neutral elements. So this is support for the idea that you get this sort of imbalance in what happens. Think about it from the perspective of threat. You get a stress reaction, amygdala plus, hippocampus minus. The outcome of any sort of a threatening situation could well be a fear memory that's not attached to context in any way. And this then is what we think connects to PTSD, where fears are generalized across all contexts and where anything can trigger a reminder. All right, what are the implications of this third story? Early aversive events create strong traces unattached to context. Adult trauma can similarly lead to fear learning, the same thing, and it could trigger PTSD flashbacks. The absence of the context link is important. A lot of PTSD sufferers, they don't just remember that this is something they relive it. The feeling of reliving is a feeling of not having any contextual link to the thing that caused the trauma in the first place. Right? So one implication, and this is being picked up now in the field, is that what you need to do in therapy is somehow recontextualize. You need to attach that fear to a safe contextual context so that in the future, any cues that trigger that memory, they will not automatically trigger fear in the same way. Some people are picking up on this. I don't know how far it's going, but that's the general idea. All right? Now, I am getting close to the end. So one could give an entire talk about memory reconsolidation. I did that last year, so I'm not going to do that now. But I want to just repeat the take-home messages. And I curiously don't really have a clock here that tells me how I'm doing, so I keep on thinking I should. All right, I'm ahead of time, which is very unusual. So we think that this approach to, let me go back one slide. Okay, the take-home messages. Sorry. Different parts of the brain mature at different times. This is the maturing hippocampus. There's a whole book on this. The hippocampus, which is critical for spatial mapping and episodic memory, matures late. Again, with little evidence of function before this time. And these are the consequences that I think I've just gone over. There's an early absence and a late emergence of exploration and episodic memory. There's an increased sensitivity to environmental perturbations. There's an interaction with chromosomal abnormalities like trisomy 21, but that's not the only one, by the way. I mean, there are other examples. It appears that most Williams syndrome and others, they all seem to have problems in hippocampus, as well as other things. But hippocampus is a common element. And any early learning that occurs is likely to show quite broad generalization. So that in the cases where some of that early learning involved fears or anxiety-provoking things, that might ultimately lead to something quite unpleasant later in life. That's the general story. And all of those things flow from the idea that this particular memory system develops later than other memory systems. And that's the main thrust of the talk. We have taken those ideas and the notion that when you reactivate a memory, as in therapy, and you try to recontextualize it, that idea that I was talking about, this involves something that is now connected to the field of memory reconsolidation. The idea that when you reactivate a memory, it creates the possibility of somehow changing it under some circumstances. So the notion now is to bring that memory reconsolidation research and the parameters that create the reconsolidation possibility into the clinic to deal with this recontextualization of a fear memory. And we're trying to push that. We're trying to do some brain research some imaging research that might shed light on it, but it's very early days. We did hold a conference on this last year, well, maybe 2 years ago now. You can see that there. There's a book coming out. I think the movie rights are still up for grabs. And this book is very much focused directly on this idea of what do we know from a neuroscience perspective, some of which I just told you here, that sheds light on how we can actually realize enduring change in psychotherapy. The big problem in therapy, one of the major problems, is they don't stick. And the trick now is to figure out a way to make it stick. Right? And so we're trying to leverage what we know about neuroscience to do that. And with that, I am going to end early, which I love to do. I have to thank a variety of collaborators. I'm only going to list the main collaborators there whose work I was talking about here. Of course, there's my co-author of the book, John O'Keefe. Jeff Wilner and I did most of the development work that I told you about back in the 80s, the late 80s, early 90s. Don Wilson did the LTP experiment. Of course, there's William James Jacobs. He's at Arizona. We still work together. And a former postdoc who did a lot of the developmental work, and my colleague on the Down syndrome and now on memory development work in general. These are some of the funding sources that I can thank for thinking I had things worth saying. And these are the fun sources that make life worth living. Thank you very much. Take a question or two, apparently. Is there a question or two? If not, yes, Nate.
I'm trying to formulate the question. So when we think about brain plasticity, we often think, or sometimes think about sensitive critical periods during which the environment has its greatest effect.
Right.
But what I understand you arguing is not that, in that the plasticity that's occurring in the first year or so of human developmental life with the hippocampus is not a sensitive or critical period, because unless you stress it or whatever, you're not going to, you're not developing the memories, you're not doing all of those. I'm trying to reconcile.
Yeah. So the question is about the relationship between notions about critical period and the story that I was telling today. And I think they're a little bit orthogonal. I mean, I don't think I was trying to leave aside the issue of, do some of these learning systems have only a window during which learning can occur in them, but then the window is shut. That's the imprinting story. But let me say something about that because it's fascinating. It turns out that the cellular molecular mechanism that controls the kind of imprinting shutoff of plasticity has to do with excuse the technical details. There are two different subunit receptors of the AMPA receptor, which is a glutamate receptor. And the balance between those two, the GLUN2A and GLUN2B, determines whether that synapse will be plastic or not. Right? There are certain conditions of that balance where that synapse is no longer going to be able to be plastic. it's been shut off plastic-wise. That happens in imprinting circuitry, and it happens in habit circuitry where the learning has been heavily overtrained. The only way to get out of that is to flip that balance, right? And that happens, so this is where the reconsolidation literature is merging with the literature, with what you just mentioned. Exactly the same subunits control whether, when you reactivate an adult memory, whether you can change it or not. If it's a heavily overtrained memory, that subunit balance has been shifted. You no longer can change it even though you've reactivated it. If the balance hasn't gone out of whack, you can still get plasticity. So there's some deep relationship between the molecular mechanisms controlling plasticity and imprinting and the molecular mechanisms controlling plasticity in general, and the hippocampus. This is every structure. This story is going to play out everywhere. It's the same, essentially, everywhere there's glutamate, essentially. So in other words, it's really complicated but interesting. And I think it's been shown in an imprinting case. Mary, you can help me here. In the vision case, that they can reopen the critical period in vision by manipulating this ratio. Yeah. So that's fascinating. Yeah. Hi.
Your talk is very interesting. I'm wondering, the hippocampus is an ancient organ, right?
That's a misconception, but go ahead with the question. Of course. Sure. That's been, the question has to do with sort of whether there's some special status for olfaction in thinking about hippocampus. And the answer is sort of yes, but nobody's really pushed. The yes is that we know anatomically olfactory input has a more direct route to the hippocampus than any other sense. That we know is sort of true, via the olfactory bulb and so on. Olfaction can get to the hippocampus faster, maybe. That's one possibility. That's one more or less fact. Other than that, there hasn't been much analysis of, of the Proustian kind of effect. I mean, are smells really better cues than anything else? I don't know that anyone's really explored that space very effectively. So I don't know if that's an answer to your question, but.
Well, as a follow-up, so you talk about the role of, the hippocampus in the ability of the organism to find things based on
Yeah.
And all my Proustian moments, of which I have maybe one or two a year, it's very non-contextualized.
Uh-huh.
Say, oh, that's a smell of coffee, or that's
Sure.
whatever. That this goes way back.
Got it. Got it. Yeah. Fair enough. I don't think I can say much about that one way or the other. Sorry. Is there another question that I might be able to answer? One more. Over here? Yeah.
You see a linkage between what you talk about fear memories that are not contextualized.
Yeah.
You see a linkage between that and the need of recontextualizing and the creation of false memories that Elizabeth Loftus.
Absolutely. Yeah, it's the same mechanisms. The same mechanisms are at play with false memory productions. Yeah. Essentially the same, because the bottom line here is that memory is way more plastic and dynamic than the field has ever admitted to. Once we admit that, then all bets are off, essentially. So these things are not fixed items the way we used to think. The whole consolidation story that I told you about was a story predicated on the notion that consolidation happens and the memory is fixed and it's over, game is over. That's wrong. We now know that's wrong. So, I mean, everything is up for grabs now in terms of memory, in terms of false memory. I mean, what is false? I mean, how well do we know when we have one of those triggers of a Proustian memory? How accurate is that? Basic science hasn't been done yet to answer those questions, to my knowledge. Anyway, thank you very much, everyone.